This Week in Microbiology is brought to you by microbe.tv and is supported by the American Society for Microbiology. To learn more about microbes, visit microbe.tv and asm.org. This is TWiM, This Week in Microbiology, episode number 339, recorded on August 21st, 2025. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from St. Louis, Missouri, Petra Levin.
It's great to be here, and we finally had a break in our horrible heat wave, and it's looks like it's about 79 degrees outside and really quite pleasant.
Yeah, same here. It's now fall, apparently, and raining. It's 21 C. Got this hurricane coming up the East Coast. Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone, and we're most grateful that the hurricane is visiting up north because last night, because of the phase of the moon, we had extremely high tides, and it was pretty spooky driving around downtown when I was coming home last night.
You mean it's north of you, the hurricane? It's past?
Yeah, the hurricane's north of us.
Yeah, it's raining here.
It's driving the surf, and, the waves are... All the undergrads are really happy because they're surfing at Folly.
Yeah, nice. Also joining us from Ann Arbor, Michigan, Michelle Swanson.
Hello. It's lovely here in Michigan, but I will say that my beloved daughter and her family are with her extended family on the Outer Banks for their traditional holiday.
Oh.
And they moved inland last night, but are back out on the Outer Banks today. So crossing my fingers that they can enjoy the holiday.
Also joining us, the TWiM adjacent, Mark Martin from Tacoma, Washington. Welcome, Mark.
Thanks a lot. I'm happy to be here, and the temperature in Tacoma is 22 degrees C.
Yeah, very similar here, but you don't have a hurricane to explain.
No, I'm delighted it's not super hot. You know me.
If you like these programs, these science programs where we talk about papers and we give you real science, no misinformation, no lies. We'd love your support to continue to produce them. You can go over to microbe.tv/contribute. We have a variety of ways you can help us out because we don't do advertising, as you may notice, because I don't like interrupting learning with advertising. It'd be like giving a lecture in a classroom and saying, this lecture is brought to you by...
Your tuition.
Yes, indeed. Your parents, they paid for it, so pay attention.
Exactly. $4 a minute, guys. $4
Sometimes,
a minute.
Is that what it is, Petra?
Mm-hmm.
Once I calculated something like that.
Pretty close. Yeah.
Oh, my gosh. And, I look at them and they're fooling around on their laptops, and some of them don't show up. And I'm like, you want me to call your parents and tell them you don't come to class? That would just not make them happy with
that.
Me,
is literally the best advice and the only advice I give to kids who are going to college. Go to class. The easiest way to learn is to go to class.
Yeah.
There's other things on top of that, but you are halfway there if you just show up.
So half of my virology class doesn't show up. So that would be like 55, 60 students. And they wait a few weeks and then they stop coming, right? I don't know why. And the others always come. The same people come all the time. And they say, that's how we learn in class. I can't learn just by watching a video, right? I have to see you say it in class and then, okay, so that's fine.
I've been to one of your classes, Vincent, and it is active learning.
Yes, you were there, Mark, and I picked on you.
Ooh.
I asked him questions. I asked him questions.
I got everyone correct.
You did. And I thought it was fun for them to see that a professor from somewhere else could come in and sit in a virology class and, first of all, be comfortable being asked questions and also get them right. It doesn't matter if you get them right or not. It's just a process, right? But they enjoyed it, I'm sure. Thank you for coming. That was great.
It was fun.
Today, we are going to talk a little bit about our departed colleague, Elio Schaechter, who passed away last Thursday, I think August 14th. He was 97 years old, and he's an accomplished scientist, had a very, very long career, which he talked about on TWiM number 200. Michelle and Michael and I went to visit him at San Diego State, and we talked to him for a while about his career, and he recounted all of his phases of his career. He spent many years. Because he was originally born in Italy, went to Ecuador for a long time, came to the U.S. and spent many years on the East Coast up in Boston, and then went to the West Coast, and that is where he spent his final years. And after retiring, he never really retired. He started a blog, Small Things Considered, and
This
came on to TWiM.
podcast.
He did this podcast. He came on to TWiM for a long time. So he joined TWiM on episode number 10. So that was June. We started TWiM in February. I asked him to join us, actually, and he had some other things to do. So he joined us on episode 10 and stayed until 263,
Wow.
which was April 2022. That was amazing. A long run. And then Petra came on to, well, he said he wanted to get a replacement, and he recommended you, Petra. So we auditioned you, and you were great. So you came on. You said, "I hope I'm not replacing Elio." No,
There
of
is
course
no
not.
replacement for Elio.
There is no replacement. Yeah, we just wanted to have another person with us. But he had a long run and had many, many different episodes where he discussed papers, and it was, his insight was terrific because he had a long experience from the very early years of microbiology when you had rudimentary technologies and watched the evolution as it went by. So he always had great insight. In fact, this blog that he writes, Small Things Considered, now is taken over by other people. But on TWiV a couple of weeks ago, we did an episode on the Meselson stall experiment. We talked about the Model E ultracentrifuge, and sure enough, Elio had written a post about it.
Oh,
And
yeah.
it was so entertaining, right? He said, what if you wanted to measure the amount of ribosomes in a growing cell? He said, today they would do PCR, but that's not right. You can't do it that way. You really need to purify the ribosomes. And here's the way we did it in the old days, which probably wouldn't make the young people happy to do it today. And he talked about the Model E. So he had that kind of insight, which I really appreciated very much. Of course, he was very productive. He did a lot of writing. He also wrote a book about mushrooms in the company of mushrooms. And of course, there's a Wikipedia page, which we will link to. He had a feature on his blog called Talmudic Questions, right? Questions that you could pose, but didn't really have an answer, but they made you think about things, which was always fun. I remember when he started Small Things Considered, which of course is an incredibly clever title, based on some news program, right?
All Things Considered.
All Things Considered, right?
And he was interviewed by the host of All Things Considered, Mara Eliason. And he got a big kick out of Small Things Considered being having him be interviewed by the program All Things Considered, which is
great. It's really good. I remember when he was going to start it, Chris Kandian helped him get the blog going, and then Ray Ortega took it over later. And his writings are really very nice. His writing style is very clear and conversational and folksy. In fact, there's a history. He wrote his own blog, his own memoirs over at the blog, which are just, this is how it starts. In one's crepuscular phase of life, and in parentheses, now that's a word I've never used before. Writing memoirs seems mandatory. It's just great how it's so folksy. And so there's a nice history of his early years. He writes, I was a lymphatic. So his mother, he was brought up, of course, in a Jewish family. My mother was a pretty poor cook, and for a Jewish mother, had little interest in food. But she decided to make him not lymphatic, to give him prosciutto. And she said, and he says, that's not allowed in a kosher household, of course, but she did it anyway. And this was like trying to waver between Jewish traditions and the desire to assimilate into Western culture. Anyway, it's really a great memoir. I would highly recommend that you read it.
And he points out that when he finally got to Ecuador, because they didn't have very much money, his parents ended up becoming vegetarians. So he was always on the scrounge for meat. He loved meat. He was on the hunt always for a good sausage. And in fact, when he was in the Ecuadorian Navy, all the food spoiled on the ship.
Mhm.
And his mother sent him into the Navy with his own personal salami. And so he was able to eat off of that salami while everybody else was literally on bread and water. And he didn't want to tell anyone about the salami. And that's one of the fondest stories I remember him telling me. And he got his first exposure, if you will, to microbiology during his time in the Ecuadorian Navy, where he cultured the fungus between the toes of Ecuadorian sailors. So he was really quite a bit of a Renaissance scientist, so to speak. He worked on many, many things.
In the TWiM 200 episode, which I highly recommend to our listeners, he described how one of his introductions to laboratory science was making media.
Oh, yes.
Now we just pull the various jars and weigh it out and mix it. He was giving a cow and needed to make heart-brain lung infusion media, something like that. Like
brain heart infusion. It was a horse.
Literally with the organs of this large animal. And he was learning by doing.
And that's actually just so crazy because if anything, the work that he did as a postdoc in Olamola's lab suggests that and the things that's been written on Small Things Considered and things like this, it's that these media that are made basically like beef broth, like brain heart infusion, vary so much you almost don't know what you're dealing with from experiment to experiment. So that's funny that he learned how to make media first. I did not know that.
Yep.
Yeah, in his biography that you can find on Small Things Considered, and we'll drop into the show notes the link for you, you can read about his experience with the dead horse from his first job as a microbiologist in Ecuador, where his boss said that the horse we were using for serum died, and they never let anything go to waste. And so consequently, they began to use the dead horse, to make all sorts of media. And I'll leave it to the listeners to discover that passage in his memoir, because it's quite a remarkable story about what parts they used, and what parts were used for.
I remember him most from TWiM because that's how I knew him. I don't think I met him before TWiM. And in fact, he did a few live events with us at ASM microbe meetings, and it's probably where I met him the first time. But he always had a really good take on papers. He would do a paper himself. It was always quite brief, right? He never went into the details, but he didn't think that was important. And then he was. I remember he was always skeptical about microbiome experiments.
Oh, yeah.
He would say, "Oh, well, this doesn't mean anything. It's just an observation. You need to do some experiments to prove what you think is going on." And I'll never forget that. That was really important. And he also, he spent years in Copenhagen, and he taught me how to pronounce it. I used to say Copenhagen. I thought I was being slick. And he said, Vincent, it's not a German name. It's Danish, and it's Copenhagen. And that stuck with me for some reason. Did you know Elio, Mark?
Yes, I did. I knew him quite well, and he was a mentor to me in many ways. He was a wonderful man, and I've done my wife and I have done several Lux portraits of them that I've shared with all of you. And I know the first one we did, I had it framed for him, and he had in his home. And one thing that you know, and I think people that have seen him lecture haven't, is he was a real character. He had a wicked sense of humor. And the first time I was at a meeting with him, we were talking, and someone who you all know that I will not name had irritated him. and I was in an elevator with him, and he used a extremely pungent curse word. And then he turned and looked at me, and the smile, and said, "Didn't think I would say that, did you?" And then he said, but blank is a blank. And I said, "Yes, sir." I will tell you that I'm not as well published as anybody here, but the most thorough editing I ever received is when I used to write entries on Small Things Considered.
Mm-hmm.
And Elio was very nice about it, but he would tell me exactly what he was thinking. He always made me welcome. He always treated me like my opinion mattered, and I miss him terribly.
Yeah, his target for his Small Things Considered post was 1,000 words, no matter the complexity of the topic, and it couldn't be turgid. He had a standard and was a masterful editor and would not accept anything short of outstanding for things that he published on that blog. He kept sending you back comments, and they were all extremely constructive, unlike some of the editorial comments people get back from manuscript reviews where they're pointless, and it's clear that they really didn't get into the paper.
No, it's true. He really curated that blog in a way that made it about the writing as much as about the science, which I think is unusual. And I mean, there's some pieces, I don't know if he edited them but there's still this piece, and going back to media by Hiroshi Nikaido, who's one of my other heroes, but on media, and it's actually talking about a paper from Dick Dari, but it's just the best description of LB and its problems, and it's on there, and it's so useful to me as a scientist, as well as to people who are just interested in these things. So it is beautiful. And I'm glad that Roberto's kept it up, and now Christoph, and it's good.
So I've got to know Elio when I was a postdoc in his department. He was at Tufts for 35 years and led the department as chair for 23. And I was there for 4 of it. And what I treasured about that time in the department is not only were there just a range of excellent scientists doing really cutting edge work, but the department was run as a democracy. Everybody shared responsibility, and they really prioritized training. So at every faculty meeting, they would run through their list of all graduate students and just get an update on each of them. So it was a wonderful community, egalitarian, and it just showed what a class act he is. I was later then privileged to join Elio and also Fred Neidhart, a former chair of my current department, and also John Ingram, in revising their textbook Microbe for undergraduates, first published in 2006. And some of the reviews of the book really capture Elio's traits that we've all already talked about. They describe its lively narrative, highly readable, and he included case studies to really bring it to life. So about how you make sauerkraut, how that's made from fermentation by microbes, the cholera outbreak in Haiti years ago. They included active learning exercises, and also used the ASM curriculum guidelines for undergraduate microbiology as principles for organizing the textbook. So it just shows you what a scholar and educator and a wonderful colleague he was.
Petra, any thoughts?
About Elio?
Yeah.
I mean, he was just always warm and welcoming. And I mean, I didn't meet him until we started to work on the relationship between nutrient availability and cell size, which is a big part of what my lab's been doing in the past 15 years. And there's a paper by Elio Schaechter, Ola Mola, and Niels Kjellgaard that I alluded to in terms of media. And this paper, and I think this goes both to Elio and to Ola Mola, it's from 1958. And I think it's in our show notes.
It is.
But it really is one of the first papers to set up how nutrient availability impacts composition, size, growth rate of bacterial cells. Because before that, people would make media. They were just interested in growing the cells and not so much interested in maybe and starting to become interested in the genetics, but not really interested in the connection between, nutrients, metabolism, and physiology. And this paper, I mean, we're interested in it for our own reasons, but everybody who does any physiology, this paper really sets up a lot of work that came later. For example, how one of the observations is that the concentration of DNA as well as protein, as well as RNA stays flat, or another way to look at it is it increases exponentially as a monotonic function of growth rate. And this means that the faster cells grow, the more of these things they have to make. And how are they regulating it? And how are they regulating RNA, right? Most of the RNA in bacterial cells goes to make ribosomes, but ribosomes make proteins. Ribosomes are also composed of proteins. So you don't want to have too many ribosomes because then you're devoting too much of your energy to making ribosomes. But you also want to make sure that you have enough ribosomes to make protein. And so there are all sorts of ideas about regulating RNA synthesis, regulating proteins, connecting them to the nutrients needed to make those. So nitrogen being a big one, carbon. And again, the paper itself is very beautiful, very simple. But it is in a way sort of like his Talmudic questions. It's just like a really simple, but so carefully done piece of work that has then spurred all sorts of other things to come out of it. The regulation, connecting nutrients to biosynthesis, and what some people, older people in the field than me, maybe call magic spot. And then people maybe my age, maybe the cutoff is 60, call guanosine or tetraphosphate or guanosine pentaphosphate. It's a GTP derivative, and how that regulates sort of the balance between protein synthesis and RNA synthesis. That kind of work really comes out of this paper to some extent. It's also influenced how bacterial size is controlled, how DNA replication is controlled. This classic paper by Helmstetter and Cooper about multi-fork replication that came after. It's actually 10 years after it comes out of that paper. Anyways, this one work has so many things and ideas that come from it, and that, it's really the quantitative study of bacterial physiology. It's really the first paper to do that in an in-depth way. And I know that Elio moved way beyond that, but he was always willing to talk to me about it. I once showed up in San Diego. I don't even remember why I was in San Diego exactly, but I made time to go see Elio. And he welcomed me to his apartment building, and we sat outside and we talked about this paper because I had results I wanted to share with him. And even though it hadn't been the focus of his work in many years, he was so patient and kind. And it was wonderful to be able to just have that one-on-one with him. So
you may have been the catalyst that caused him to write his 2015 paper that appears in Frontiers in Microbiology on the dawn of the bacterial physiology era. And it, along with 30 other papers, really sets the tone. And Elio reviews the dawn of bacterial physiology and that really elegant review that he wrote just 10 years ago when he was 87. And one of the quotes from it, and Elio writes, and I'll just quote him here: "The importance of growth at a steady state has been realized earlier, but Campbell's novel and precise term helped remove the aura of immutability from the growth curve. It provided the freedom to manipulate cultures by, for instance, repeatedly diluting them so as to maintain them in balanced growth. One of the most interesting of these manipulations in the early 1950s was the development of the continuous culture in chemostats that was pioneered by the father of molecular biology, Jacques Monod."
Yes. So that's a really important concept, because before sort of the work by Olamola and, that Schaechter has been associated with that Elio has been associated with through this 1958 paper, people were not that careful. They knew there were different parts of the growth curve, but they weren't that careful. And understanding physiology, you really need to be sure not only are you replicating the media, but you're replicating what stage of the growth curve you're at. So like You can do that by chemostat, which essentially keeps things at the same optical density by adding fresh media and taking away less fresh media. Or you can do it by repeated dilutions and being careful about which optical density you sample at. But I think a lot of people who do physiology, it's kind of at your peril that you don't pay attention to that, because like E. coli starts to go into stationary phase. It starts transitioning before it even gets like around an OD of 0.5 OD 600. And, some bacteria are later, but that's E. coli. And so if you're above that, your cells are already changing their gene expression, and as long as you're consistent. But I think that's kind of the message, and that's the message of that 2015 review paper. Actually, June, I think it's 2019, has a really beautiful, very detailed history of bacterial physiology and the relationship between quantitative micro and physics, in which all of these people figure really strongly, including Elio Schaechter, Ola Mola, Alan Campbell. That's in, I think, a physics journal, but it's by June, and I'll put that up to link it. But if you're interested in this history, so there are really two schools, though. So there's the Ola Mola, study the physiology really carefully, don't perturb the system, don't put mutations in. And then there's so that's sort of sometimes called the Copenhagen school. And then there's this other, Jacob, Monod, people who came after, that is interested also in mutations. And making mutations to understand. And so these two schools, and then you have the phage school, right? The people who figured out a lot of the molecular biology by studying bacterial phage, invasion, and things like this. So you have these different kind of forces coming together, but they kind of all agree in some level. Their conclusions are sometimes pretty different, but in terms of the rigor and the reproducibility and how to understand the bacteria and as an organism in and of itself, I think really comes together kind of in this, how we grow them, how we think about them, and how we kind of can compare between different experiments.
Any other thoughts about Elio before?
Yeah, in addition to his, starting out with taking a horse and turning it into growth media, and then his amazing quantitative experiments that have just been described. As we mentioned, he was chair of a department. He also was on the board of the American Academy of Microbiology. He served as president of the American Society for Microbiology. And if that's not enough, he also not only wrote that undergraduate textbook, but also published a textbook for medical students, Schechter's Mechanisms of Microbial Disease, plus his book on his love of mushrooms, the podcast, the blog. So I think he's just such a great role model to show us just how many opportunities there are available as for a microbiologist to contribute to the field at different stages of life and different phases of your career.
One last thought. He often quoted the maxim of the Spanish philosopher Jose Ortega y Gasset when describing bacteria behave the way they do, and quoting, "Yo soy yo y mi circunstancia," or in English, I am my circumstance. Yeah. So to all of you, Elio, it's been a journey.
I just wanted to say that Elio is one of the people that I wanted ASM to follow around with a recorder for about the last 10 years. And the reason why is so many folks that I've known who, I'm not going to say a generalist, the polymath. And Elio was so good at looking at problems that were developed in the last 10 years. And he understood what they were about. He understood the positives and the negatives and put them into context. And I think we miss that because we get overly specialized. So he's one of the people I wanted. I promise you, forgot more microbiology than I will ever know. And I love me some microbiology.
Do you, Mark?
Yeah, I do. I'm wearing the microbial evangelist button.
I know you do. I was going to give you an intro to your paper.
Please do.
I was going to say, so if you like microbiology so much, tell us whether archaea and bacteria compete with each other, Mark.
Well, it's really an interesting concept because when you think about it, Vincent, there was a period of time when the microbial world was thought of, call it in a Margulian way, where everyone got along. And I was always very uncomfortable with that because, what we want to be true is not necessarily true. So kind of like, I think it's now come to the forefront, and this paper is a great example, that it's all about conflict. And if we can say red in tooth and claw, perhaps we need to change the microbial world is red in pilus and EV, right? Because that's really what we're talking about here. And this is a fabulous paper. And I want to say something to start with. I get into trouble every single time I say that I wish there was an E. coli among the Archaea. And everyone comes at me with sulfolobus and halobium and halovorax, and those are all great. But I think the problem is the way it's taught. We don't get enough exposure to the archaeal world, how common and diverse they are. We just tend to focus on the extremophiles or the anaerobes. And the title of this paper is Archaea produce peptidoglycan hydrolases that kill bacteria. Now, I've looked at a lot of papers that, beat around the bush. I love that title. And this particular paper came out in PLOS Biology. The authors are Strock, So, Misson, oh my goodness, Romalotai, Shilaha, Hochner, and Tobias Warnecke. And these are all folks in the UK at Trinity, Oxford, the Medical Research Council Laboratory. It's a really good group. And it's a fabulous paper to read. And I want to say it came out in PLOS Biology on August 14th of this year. And speaking of Elio, our late friend Elio, he really pushed on me the depth and breadth of sociomicrobiology. And too many times people say that's just about quorum sensing and the formation of biofilms. He would always gently correct me on that. It's a larger concept. The idea that microbes don't live in monoculture, but as diverse communities. And so there are all different kinds of relationships, from syntrophy to the predation that I love from things like Della Vibrio. Competition is the rule. And as I say, the microbial world is red in tooth and claw, or in pilus and EV.
Yeah, Mark, can you just tell us what's the timeline for emergence of bacteria in Archaea?
Well, this is a religious argument in a lot of ways, Vincent.
And while you're at it, tell us whether oxygen or nitrogen was fixed first, and then we'll.
You really are going down the rabbit hole now, Petra.
Nitrogen was fixed first. Come on. No, this is wonderful. I really appreciate it because I'm going to be teaching here in a couple of weeks. So I love doing that to people. Petra, by the way, it's a great question to ask students. What's the most important enzyme on Earth? And they invariably go to DNA polymerase.
Oh, no.
Elio used to say Rubisco.
It is.
Yes. Or nitrogenase. Or nitrogenase.
Yeah.
Absolutely.
Or nitrogenase. Rubisco or nitrogenase.
Huh.
The most abundant is still Rubisco, though, right?
Yeah.
Yes.
For now. We're trying to make sure that we've destroyed all those things, but that's another story.
Back to the paper.
Yes.
No, but I don't. But so you can't tell me who was first on Earth, right?
The answer is yes. And what I mean by that is I don't think there's strong evidence. If you look at the molecular biology of bacteria and archaea today, I can't bring it back to Luca very well. And when you look at the last universal common ancestor, people often will say, well, they're all hyperthermophiles. And I don't know if that's true or not. It's a really hard question to answer. And this takes me back to what I started with saying, archaea, we tend to focus on the extremophiles. We should be looking at other ones too.
But to answer Vincent's question, they both have been around for about 3.5 billion years.
Yeah.
So there's a lot that we don't know that happened in between then and now.
Yeah, I mean, the question is interesting because here we're going to see they exist in many environments.
Yes.
Not right. And what they say in this paper, bacteria outnumber them greatly. But that kind of suggests that they arose contemporaneously rather than having, say, bacteria established and then the archaea having to find niches. It's less likely that they would overlap, right? That's why I ask you that.
No, and I'm more and more convinced that eukaryotes are archaea that have incorporated bacteria, even though there are some issues with that. So to get to the paper, to anticipate Michelle, and thank you, Michelle. I mean that genuinely. Uh, they wanted to look at something that had not occurred to me. If you're going to talk about competition, what they wanted to show is that could they look at enzymes that an archaean could make that would impact bacteria. Now, there are particular proteins called halicins that are made by many archaeans that affect other archaea. Some of them are pore formers. That everything's a. My students always say everything's a pore former. This is not always true. So, what they decided to do is ask this question, taking us back again to Elio: what do bacteria have that archaea don't? And it's our good friend peptidoglycan. And though it is true that archaeans don't make peptidoglycans, some of them, they have to make a molecule that serves the same function, which I'll get to. So what they did is they wanted to know if archaeans actually made things that could break down peptidoglycan. Parenthetically, bacteria having peptidoglycan will make enzymes that break down peptidoglycan, often called autolysins, but this is because they have to snip it a little bit in order to build on it. And just for folks who were new micronauts, I want to say what peptidoglycan is a heteropolymer of N-acetylmuramic acid and N-acetylglucosamine. I teach my students to say NAM and NAG, and that's the glycan part of peptidoglycan. And there are short peptide bridges giving them a mesh and strength. Those peptide bridges are cool beyond words because they often include D amino acids, which is fantastical. So this mesh helps protect them against environmental insults like change in osmotica, okay? And everything needs something like that to protect against that. So what they decided to do then, and by the way, I shouldn't say this, but I will. I tell my students something that haunts their dreams. I tell them that peptidoglycan is like Spanx for bacteria.
Mm-hmm.
And they'll never, ever forget it. Okay? So what they decided to do, and they have a problem here, how do you go looking for these things? And so what they wanted to do is focus on enzymes that break down peptidoglycan, again, produced by archaeans, and they used a predictive structural homology approach. And then, again, back to Baylor, they did experiments. So they decided to focus on, and I'm getting ahead of myself a little bit, on the archaean halogranum and the bacterium Halactobacterium. Now, these are both from saline environments. What I really want everyone to do is have a look at this paper and read the introduction, which is written in such a fabulous style, discussing the rules of engagement between microbes in a given environment. And prior work had been done for many, many years showing antagonisms by using culture supernatants to show inhibition of a target microbe. But are these effects specific or general? So they decided to look for enzymes that break down peptidoglycan. They used a genomic search to do that. And again, archaeans are very different in a wide variety of ways. So it's a clear target for antimicrobial activity, and the authors give several examples from bacterial interactions and archaeal interactions. They actually mentioned a little bit, but again, I want to emphasize some of these enzymes are present in bacteria, and that might confuse a new reader saying, why would they make an enzyme that breaks this down? Plenty of examples in biology of polymers that need to be nibbled back. So they started to go looking for this, and they found out that the enzymes that do this, they're called peptidoglycan hydrolases. Some of them, and I love this expression, have been weaponized by some bacteria. And that's a fascinating idea. Turns out that PGHs, peptidoglycan hydrolases, excuse me today, I already did one podcast. They're modular, and this is very helpful. They have the module or the domain that binds peptidoglycan, and then they have a catalytic module or domain. Now, if you look at the Staphylococcus, they have enzymes like this with a secretion signal, and they're the weaponized ones. The authors looked at 3,700 archaeal genomes and 50,000 bacterial genomes. Now, obviously, lots and lots of bacteria have PGHs, but 5% of the archaeans seem to have them too. And 32% of those have secretion signals. So they make an enzyme that could be secreted. If you look at figure 1, there's, and I'm not being critical. I have such respect for the authors, but when figure 1 has like 8 different things in it, you have to kind of like just look at it carefully. Sometimes the legend runs on to the next page. But what they show you is the relationships between the archaeal and bacterial pHs in terms of their abundance. And it very clearly, there are many, many bacteria and quite a few archaeans that seem to have these. Clearly, horizontal gene transfer has been in there. And so because I know we're watching time very carefully, and I want to respect that, I want to tell you that they decided to focus on archaeal homologs of something called zoocin, Zoocina, which is the best described weaponized peptidoglycan hydrolase in bacteria. Turns out when you look at all of those different homologs, you see lots of evidence of domain shuffling. So it's one of those things I often tell students is that genomes are plastic. They're constantly changing. And they wanted to kind of look at it because they think of it as being an arms race regime, that the peptidoglycan hydrolase utility changes rapidly as the targets adapt and new targets emerge, and so you have gain and loss. So what they did, I hate saying cut to the chase scene, but I say it all the time. In halogranum, one of the archaeota that they're looking at, they found a homolog of I want to say zoocina, but it's zoocina, and they named it, and you have no idea how much I adore this, Waldo.
Mm-hmm.
That's the moon blade from historical Korean weaponry. Later on, they found a longer version of it, also in halogranum, and they call that Dan Waldo, which is larger still in terms of ancient weaponry.
Mm-hmm.
So when they had that, and again, it's got a secretion signal on it, they used the binding module to make predictions of possible targets. Because it isn't enough just to look at the sequences. They wanted to show that things were really happening. And one of the possible targets was hal alkali bacterium halodurans. And they were able to show that culture supernatants appeared to inhibit from the archaean. And so they cloned the Waldo gene into the halopharax system. And they did a beautiful job for first-year students showing with and without the insert, supernatant inhibits, supernatant doesn't. And they were able to further show that Waldo is as bacteriostatic. And it's not just, excuse me, I meant to say bactericidal, because a lot of antibiotics or antimicrobials simply go after things that aren't growing. But they checked CFUs and did live dead staining. They also did some mass spec to confirm it. So what they ended up doing is they showed that a diverse amount or diverse number or diverse array of archaea encode these peptidoglycan hydrolases. And they target the cellular structure that's unique to bacteria, peptidoglycan. At least 3 of these archaeal peptidoglycan hydrolases, and it was Waldo and Dan Waldo from the one archaean, and then another that they found that they just found in the process from another archaea kill bacteria. Now, this is interesting because normally when I say that microbes duke it out for space and resources and that kind of thing, we're now starting to learn that there's a lot of fighting that takes place one way or another. And that's what we're looking at here.
So let me ask you a question, Mark.
Yeah.
So the Waldo and Dan Waldo, if I was an executive for a pharmaceutical company, could these 2 molecules be adapted to become targeted antimicrobials against multi-drug resistant bacteria?
This is the thing that I would try and do. And if you think about it, you would have to do maybe some in vivo evolution of the peptidoglycan binding area to make it specific. One of the problems all these things have, and it's interesting to me, I want to know how tight that association is to that module. And I don't know this for a fact, but I would be greatly surprised if folks weren't paying to do that, develop a company right now.
Mm-hmm. Well, one of their really cool experiments that you may get to in a second is what they did with Waldo. I found it absolutely fascinating when they broke its catalytic domain and it had no effect.
Yeah.
That was a good
So
day in the
it-
lab.
Yes.
That clear result.
Yeah, and the pictures are pretty clear on this. And so, I was looking at that thinking it's just gumming up the ability to make more peptidoglycan, right? And that's just one thought. But as you say, I mean, it seems quite specific. There are lots and lots of bacteria it doesn't work on, which is very interesting to me.
Well, it's good for the whole notion of antibiotic stewardship and using only antibiotics against pathogens that are causing patients disease rather than taking out our entire microbiome like most antibiotics do.
So specificity is good.
Specificity is outstanding.
I couldn't agree more. I mean, one of the things I worried about with Delavibrio as a therapeutic agent is that it's quite a generalist when it comes to gram negatives. Although there's some new reports out on that are really exciting. But like bacteriophages, we can hopefully learn to direct these things.
So the question is, what's giving them specificity?
I agree.
In the generic description of peptidoglycan,
yes,
it's not immediately clear. Bacteriophage need receptors, right? So that specificity makes 100% sense.
So it makes me wonder what's special about that particular binding site. And just as some enzymes can recognize specific nucleotide sequences, I wonder if they can recognize elements of maybe the conformation of peptidoglycan that aren't evident just from the primary structure of the polymer.
Right. And usually if there's variation in peptidoglycan, it's not the NAGNAM, it's not the glycans, it's the peptides.
Mm-hmm.
Yeah.
Those vary a lot. And then, so that's actually what's my thing. I'm like, because usually like lysozyme, is active. It's not active against staph peptidoglycan are very good. So you use lysostaphin. It's a hydrolase, but it works against E. coli and bacillus, which are very divergent organisms. So
I'm really happy to hear you say that because so often people will tell me a simple explanation for something that's quite complex. And they'll simply say things like gram positives have such a thick layer of peptidoglycan. And then I tend to like to ask them, and I learned this from Elio, how do you know that? What's the experiment? When you read it's 50 layers thick, who did that? Right? It's a really interesting question. Is it thicker than you find in gram negative? Sure. Right?
But the layers, right? I don't know how they know how many layers. I know by TEM it's really obviously thicker.
Yeah.
But then the differences in these anything that attacks peptidoglycan as a target, like the specificity is where I am always a little confused.
So their data point out that there's something we don't understand about
exactly. Right.
So that's
exactly.
Beautiful, right? That's a real contribution.
Michelle, I'm interested in how, and I don't know the literature on autolysins, but I wonder about the level of specificity in autolysins, and that might be another way of looking at this problem. What do you think?
It seems reasonable.
Yeah. So again, I want to watch our time. Yeah, we're almost there. The end of this paper. Oh, first off, the materials and methods are great. You got to read those. Even the stuff about mass spec and all the rest, which okay. More power to that technique. I want to see like on a plate. I want to see culture supernatants and stuff like that. But they actually have a really nice summary. And I'm so delighted to see this because when I teach about MAMPs in class, when we talk about recognizing symbionts or innate immunity, I'll say, well, what is the specific signal that there's a bacterium? And one of them, in fact, is peptidoglycan, okay, or a fragment of it anyway. So in the end of this, they kind of break things down into 4 points in their discussion. And the first thing they tell you is there are other things than peptidoglycan to look at if you're interested in how Archaea can interact with bacteria. And again, we tend to think of things simplistically with an antagonist and a victim. But in fact, there's a whole universe. And they actually talk about this a little bit, having to do with some of these saline-loving microbes, that some of them are found in areas that change a lot, say as the tide goes in or goes out, or fresh water comes in or goes out. So maybe this is one arrow in their microbial quiver to help gain an upper hand among the archaeans. One thing they started to think about, look at the lipids that are used in archaeal membranes versus bacterial membranes. So maybe, in fact, they can look at that to go after the ester bonds instead of the isoprenoid chains linked by ether bonds. And again, looking for archaeal genomes for enzymes that specifically interact with bacterial phospholipids. That seems quite general, but at least it's the same idea. Second, they say we don't know all of the potentially diverse methods by which archaea kill bacteria, but we do know a lot about how bacteria kill bacteria. And they say, let's look at those homologs. And I'm starting to get nervous when they say systems homologous to known bacterial weaponry. First off, points for saying bacterial weaponry. But then they say this, many archaea encode contractile injection systems, like we see in type 3s and type 4s. And those are used to kill other archaea, but whether those same systems can be used to target bacteria remains yet to be tested. But again, they actually say elsewhere you have to be very, very careful with these homologs, because you there's a lot of domain shuffling, a lot of confusion at that. So they actually did look for homologs of bacteriocins, and they were able to show that some of the bacteriocins that are found, like, for example, in the lactobacilli, are never found in archaea based on their sample size, but others are found in archaea, like colicin M and saccharine A. And so you might be wanting to look for archaeal-produced bacteriocins, but it's again, you have to be careful. The link is less specific because proteins and small metabolites could be active against bacteria but evolved to fight against archaea. So you have to be kind of careful. Now, they do say something that I'm a really big believer in. If you're looking for homologs, you won't find anything fundamentally new. And I believe that. And I've seen that over and over again in science. But I do think this is a great place to start. And I'm really happy to have seen it. And then they...
Where are you going, Mark? Are you leaving us?
No, am I gone?
You said, I'm really happy to have seen it. That was a funny statement.
No, this is a great paper.
Yeah, no, you're happy to see the paper, not us. Maybe you're happy to see us.
I am delighted to see, and I'm here, Michelle, of course. But what's interesting to me is they make the comment, and again, I worry about making assumptions. In bacteria, a lot of phage defense proteins are linked together. They're in clusters, and they wonder if the same thing's true for antibacterial weaponry in archaea. And that could be. But again, this is just they're trying to kind of get people thinking about this. But I'm so happy that they say in the final analysis, doing culture experiments is really the most promising thing to do.
Mm-hmm. Yeah,
I think this is a wonderful paper for people to think about how to analyze a complicated system.
Right. I appreciated that they ended with this term, a roadmap, and really laid out their thinking and what outstanding questions there were. And also that they put out the very provocative idea that perhaps this is a great way to look for new specific antibiotics, antibacterial.
Yeah. And they're cautious about it, Michelle, and I'm glad they are, because, do you get anything new? But that's not really what's important here, because you want something where the target is unique to bacteria.
Yep.
So if you can find anything at all that binds to something bacterial, and then link that module to something that pops the cell, that's important.
This is a relatively good sized protein, right? So therapeutically, it's a little challenging.
Right.
Oh yeah. And I mean, and this is in this very unique system that I don't think would work with people at all. So it would have to be adapted to be less extremophilic.
So could you do a co-culture with bacteria and archaea be able to grow together in the laboratory? I know in nature they do, but
yeah.
Have people done that?
And you have to use halophilic bacteria for it because the Archaea that grow best at the temperatures and pH conditions of bacteria are going to be the halophilic ones.
Wouldn't you want to look at the microbiome and looking at the methanogens and the true bacteria in our gut?
I think that that's a really good thing to start looking at. And I didn't dig into the phylogenetic clades they put up there to ask that question. But that is part of their data set.
And I'm sure when our friends in the pharmaceutical industry begin to dissect this paper in their lab meetings, they will most definitely look at the opportunities, especially for those extremely multi-drug resistant gram negatives that is plaguing medicine today.
Yeah. And I'm also interested, though, in this idea, now that I think about it, in the gut, like how there are these kind of symbiotic relationships.
Mm-hmm.
There's Syntrophomonas, and I think it is a methanogen, right? That it can't, they have to work together. And I'm wondering if this is also a selective mechanism for some of these relationships.
Well, the big question that students always ask me is, how do you maintain the proper population structure in the gut? And this finding this weaponized business, might be applied to interlopers into the proper associations that we need to see. But I think that would be a fabulous thing to start looking at, because we know the gut there's lots of phages. We know that. We know there are lots of extracellular vesicles carrying Lord knows what. So why
and
not
then the
these?
Immune system is also selective in some way. And then, but you do need to maintain the structure for the ecology of the system to work, right? For the degradation pathways to work, you have to maintain this super strict structure. Essentially, each one is acting as a sort of little enzyme. If you think of bacteria as an enzyme to degrade what we eat, or in any microbiome, and you have to maintain the right proportion of those things, or you're going to go too far or not far enough. And so I'm wondering if some of that, the ecology is regulated by some of these.
And we see that in ruminants, where they will get an overgrowth of particular microbes that disrupts everything.
Yeah,
just
it's a chemical reaction just on a macro scale, micro-ish scale.
It's like brewery syndrome, right?
Exactly. Where they got to degas the cow.
Don't want to be around for that one.
No, it's not pleasant.
Thank you, Mark.
You're very welcome.
That's very cool.
That's TWiM 339. You can find the show notes at microbe.tv/TWiM. Questions, comments, TWiM@microbe.tv. If you enjoy these programs, we'd love your support. Microbe.tv/contribute. Our guest today from the University of Puget Sound, Mark Martin. Thank you.
You're welcome.
Always good to see you and your surroundings. Michelle Swanson's at the University of Michigan in Ann Arbor. Thank you, Michelle.
Thank you. It was wonderful to share our remembrances of dear Elio.
Michael Schmidt, Medical University, South Carolina. Thank you, Michael.
Thanks, everyone.
Petra Levins at Washington University. Thank you, Petra.
Thank you. And it was really nice to be able to share our memories of Elio. And in Judaism, we always say, may his memory in this case be a blessing. And I think all of us were blessed to have known him. Mm-hmm. And his memory will continue to bless us.
I will eat mushrooms for him this evening.
Just make sure you cook them, Mark.
Yes, sir.
And pick the right ones.
Yeah. He's the one who told me there are old mushroom hunters and bold mushroom hunters, but there are no old, bold mushroom hunters.
That's funny.
And on that note,
that's great. I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. See you next time on This Week in Microbiology.